this sequence that we are discussing today is referred to as the central dogma. The central dogma is an explanation of the flow of genetic information within a biological system, specifically dictating that DNA is transcribed into RNA, which is then translated to make proteins. So let's say we are looking inside of a T cell within a eukaryotic organism, all right? A T cell is a type of white blood cell that contributes to your immune system. So let's say this T cell needs to produce cytokine proteins, which are proteins that help regulate the activity of the immune system. How will the cell do this? Well, it will all begin in the nucleus of that eukaryotic cell. Here we have our directions in the form of DNA. DNA is too large to leave the nucleus, and the cellular machinery used for reading genetic code and using said code to make proteins, aka ribosomes, are out in the cytoplasm of the cell, right? They're outside of the nucleus. Therefore, DNA must be transcribed into RNA so the directions to make cytokine proteins can reach the ribosomes outside of the nucleus. Remembering back to Unit 0 material, RNA sequences are much shorter than DNA, and if we are transcribing just a small part of the DNA onto mRNA, then of course that will be a much shorter bit of coded information to transfer. So that will be small enough to leave that small nuclear pore in the nuclear membrane. This transcription process, transcribing DNA into RNA, takes place in 3 steps: initiation, elongation, and termination. All genes, including the gene that codes for the cytokine proteins, have something called a promoter. As the name would imply, promoters help promote transcription by essentially marking where a gene begins and where proteins should bind to the DNA to initiate transcription. This is the first part of initiation. In eukaryotes, promoters are a sequence of DNA that include a sequence called a Tata box, which reads TATAAA, right? Thymines and adenines, along the coding strand of DNA, and is easily recognized and bound to by transcription factors. This promoter/Tata box is found just upstream of the gene in the DNA sequence that we want to transcribe. Initiation is when an enzyme called RNA polymerase binds to the promoter. This signals the DNA to unwind, so the RNA polymerase can read the bases in one of the DNA strands. Now, I know you just heard the word enzyme, so immediately some of you might feel like we're facing a sort of chicken or egg situation here. Yes, we need proteins, such as RNA polymerase enzyme, to make proteins. So how did we get the original proteins that help us make more proteins? Well, as it turns out, these essential proteins that help with gene expression are actually gifted to us by our biological mothers. So in addition to their DNA, a biological mother's egg contains these essential proteins for gene expression. Thank you, moms. Okay, so RNA polymerase has bound to the DNA, and the DNA has unwound. The enzyme is now ready to make a strand of mRNA with a sequence of bases that are complementary to the original DNA strand, which brings us to step 2 of transcription, elongation. Elongation is the elongating or addition of nucleotides to the RNA strand. RNA polymerase reads the unwound DNA strand and builds an RNA molecule using complementary base pairs, where C's pair with G's and A's pair with U's. As it turns out, RNA does not have thymine. Instead, it has something called uracil. So during the elongation process, an adenine in the DNA is paired with a uracil, or a U, in the RNA. Now, RNA polymerase only works on one strand of DNA. So to help keep the two strands of DNA straight, we refer to one strand as the sense strand and the other strand as the antisense strand. So when you hear those terms, this is all they're referring to, right? The strand that is transcribed by RNA polymerase is called the antisense strand, and it is complementary to the RNA sequence that's being built. The strand that is not transcribed is called the sense strand. If RNA polymerase carries out its job correctly, the sense strand should be basically identical to the RNA sequence that's being made, except there are U's in the places of where T's were in the DNA sequence. Make sense? So once we have our RNA strand, the next step is step 3, termination. Termination is the ending of transcription, and it occurs when RNA polymerase crosses a stop or quote-unquote termination sequence in the gene. The RNA strand is complete and it detaches from the DNA. DNA has been successfully transcribed into RNA, meaning the directions are copied and almost ready to leave the nucleus. Yay! So, circling back to our original scenario, within the nucleus of our T cell, the gene for a cytokine protein was read and transcribed into RNA by RNA polymerase. Now we have a copy of the directions for making those cytokine proteins that are small enough to exit the nucleus through the very small nuclear pores. But before the RNA can leave, it must undergo some modifications, because once it leaves the nucleus, it's going straight to the ribosome to be translated into proteins, and we need to make sure it's ready for the journey.